From the moment we take our first breath to our final exhale, our bodies are engaged in a delicate dance of gases. Oxygen, the vital elixir, flows into our lungs, eager to fuel our cells. Meanwhile, carbon dioxide, a waste product of our metabolism, makes its exit. This exchange, this intricate ballet of inhalation and exhalation, is the essence of respiration. To truly understand this fundamental process, we must delve into the fascinating interplay between oxygen, carbon dioxide, and the protein that ferries them through our bloodstream, hemoglobin. Hemoglobin, residing within each red blood cell, acts as both an oxygen taxi and a carbon dioxide shuttle. But its cargo-carrying capacity is not static. It's influenced by factors like acidity and the concentrations of the very gases it transports. It's here in this dynamic interplay that we encounter two crucial phenomena, the Bohr effect and the Haldane effect. Imagine our hard-working hemoglobin molecules diligently transporting oxygen from the lungs to the tissues. As they reach their destination, they encounter an environment teeming with carbon dioxide. This abundance of CO2 increases the acidity, lowering the pH. And here's where the Bohr effect takes center stage. The Bohr effect describes how hemoglobin's affinity for oxygen decreases in acidic environments. Think of it like this: in the presence of high CO2 and low pH, hemoglobin becomes a less enthusiastic oxygen carrier, readily releasing its precious cargo to the tissues that desperately need it. This effect is particularly important during exercise. As our muscles work harder, they produce more carbon dioxide, further enhancing the Bohr effect and ensuring a steady supply of oxygen to fuel their contractions. Now let's turn our attention to carbon dioxide, the byproduct of cellular respiration. Just as oxygen relies on hemoglobin for transport, so too does CO2, albeit through a different mechanism. Enter the Haldane effect. The Haldane effect focuses on how oxygen influences hemoglobin's affinity for carbon dioxide. In oxygen-rich environments like the lungs, hemoglobin has a reduced affinity for CO2. This means that hemoglobin is more eager to release carbon dioxide, allowing it to be exhaled from the body. Conversely, in oxygen-poor environments like our tissues, hemoglobin's affinity for CO2 increases. This facilitates the uptake of CO2 from the tissues, where it's produced, to the lungs, where it can be expelled. The Bohr and Haldane effects, though distinct, are intricately linked, working in concert to optimize gas exchange. The Bohr effect ensures oxygen delivery is enhanced where it's most needed in metabolically active tissues. Meanwhile, the Haldane effect facilitates the removal of carbon dioxide, preventing its accumulation and ensuring the smooth functioning of our cells. These two effects highlight the remarkable adaptability of hemoglobin, a molecule that constantly responds to the changing demands of our bodies. Whether we're sprinting towards a finish line or simply enjoying a quiet moment, the Bohr and Haldane effects are silently at work, ensuring our cells receive the oxygen they need and are relieved of the carbon dioxide they don't.